Assalamualaikum warahmatullahi wabarakatuh. My name is Nily Su'aida. I'm postgraduate student from Faculty of Pharmacy at Langga University. Today I'll be presenting about the vaccine development from preclinical studies to clinical trials. So what is vaccine? The word vaccine originates from the Latin variola vaccine or cowpox, which Edward Jenner illustrated in 1798 could prevent smallpox in humans. Today, the term of vaccine applies to all biological preparation produced from living organisms that enhance immunity against disease and either prevent, we call it prophylactic vaccine, or in some cases, treat disease, and we call it therapeutic vaccine. Vaccines are administered in a liquid form, either by injection, by oral, or by intranasal route. Vaccines are composed of either the entire disease-causing microorganism or some its components that may be constructed in several ways. And there is the type of vaccine. The first one is live attenuated vaccine. It forms living organisms that have been weakened, usually from cultivation under suboptimal conditions, also called attenuation, or from genetic modification, which has the effect of reducing their ability to cause disease. And the example of this type of vaccine is measles, mumps, rubella, and varicella zoster. Number two is inactivated. So this is from whole organism that have been inactivated by chemical, thermal, or other means. And the example from this vaccine is hepatitis A, influenza, pneumococcal polysaccharide. And number three is recombinant subunit. This is from components of disease-causing organisms, such as specific proteins and polysaccharide or nucleic acid. And the example is hepatitis B. Number four is toxoid vaccine. It's from inactivated toxin of toxin-producing bacteria. And the example is tetanus and diphtheria. And the last one is conjugate polysaccharide protein. This is from the linkage or conjugation of polysaccharide to proteins. This increases the effectiveness of polysaccharide vaccine in young children. And the example is a pneumococcal, meningococcal, and Haemophilus influenza type B. And how vaccine works. To understand how vaccine works, it helps to first look at how the body fights illness. When germs such as a bacteria or a virus infect the body, they attack and multiply. And this infection, called an infection, is what causes illness. The immune system uses several tools to fight infection. Blood contact red blood cells for carrying oxygen to tissues and organs, and white or immune cells for fighting infection. These white cells consist primarily of macrophages, B lymphocytes, and T lymphocytes. Macrophages are white blood cells that swallow up and digest germs, plus dead or dying cells. The macrophages leave behind parts of the inviting germs called antigens. And the body identifies antigen as dangerous and stimulates antibody to attack them. For the lymphocytes are defensive white blood cells. They produce antibodies that attack the antigens left behind by the macrophages. And there's also T lymphocytes, are another type of defensive white blood cell. They attack cells in the body that have already been infected. The first time the body encounters a germ, it can take several days to make and use all the germ-fighting tools needed to get over the infection. And after the infection, the immune system remembers what it learned and how about to protect the body against that disease. The body keeps a few T lymphocytes called memory cells that go into action quickly if the body encounters the same germ again. And when the familiar antigens are detected, the lymphocytes produce antibody to attack them. So there is establishment of vaccine specification step. The first one is to identity. The purpose is to show that the vaccine contains the intended immunogenic component. The second one is purity or impurity. The purpose is to identify and characterize any component present in the product that is not desired product. An impurity may be either process or product related. Number three is potency. This is to estimate the biological activity and immunological strength of a vaccine. And number four, there is stability to demonstrate that the product vaccine is stable in the final container and to set the shelf life under appropriate storage condition. And number five is viral safety testing. This is to identify and control viral contamination of the cell banks, viruses, or vaccine bug that may arise from the original source of the cell lines or from the adventitious introduction of virus during production process. And the last one is transmissible spongiform encephalopathy agent. The purpose is to minimize the risk of transmitting animals spongiform encephalopathy agent via human and veterinary medicinal. And this is the figure that show the process of preclinical and clinical study and also manufacturing from product process and formulation development. One of the most challenging aspects of vaccine design remains assessing the efficacy and effectiveness of new vaccine formulation. The ultimate test of vaccine efficacy is, of course, protecting humans. But when choosing which vaccine candidate to take into clinical trials, other surrogate approaches are needed. The critical points along the preclinical pathway include detailing the host pathogen interaction, understanding the protective immune mechanism involved, and selecting appropriate antigen and adjuvant to achieve the desired immune response. Subsequent steps involve the production of the antigen and compatible vaccine delivery system, followed by development of immune readouts and toxicological tests to assess the safety and performance of the candidate vaccine construct under preclinical evaluation. And this is a preclinical safety assessment study design. So preclinical testing is a prerequisite to move a candidate vaccine from the laboratory to the clinic and includes all aspects of testing, product characterization, proof of concept, immunogenicity studies, and safety testing in animals conducted prior to introducing the product into humans. And also toxicity studies should be designed to mimic the intended use in the clinic. Thus, the relevant animal species or strain, dosing schedule, and method of vaccine administration, as well as timing evaluation of endpoint should all mimic the intended clinical use. Special consideration should also be given to the quality of the lots of antigen to be used in preclinical study, as they must be representative of the final vaccine formulation. And those a panel of toxicity studies is required by the regulatory agency because such as a single dose toxicity, repeated dose toxicity, and safety pharmacological to address this question. The goals of these studies are to provide information on systemic toxicity and local tolerance, and also identify potential target organs for toxicity and assess whether such toxicity is reversible, and identify safety parameters for clinical monitoring and help plan protocols for subsequent clinical studies in human subjects, and also identify initial safe dose and subsequent dose escalation schemes in humans. And the next step after the preclinical safety is clinical evaluation of vaccine. The goal of the clinical development plan is to demonstrate a vaccine safety, immunogenicity, and efficacy. There is a 4 paths for clinical evaluation or clinical trials. With Path 1 trials are intended to provide a preliminary evaluation on the safety and immunogenicity of the vaccine candidate in a small number of closely monitored adult volunteers. Usually, these Path 1 studies are blinded, those ranging placebo-controlled studies involving 20 to 80 subjects, and they may last up to 1 year. And during the Path 1 study, various composition or formulation of the vaccine candidate may be tested. And Path 2 trials, the purpose is to aim to provide further information on safety and immunogenicity and data on doses and schedule of the vaccine candidate that will be used in the Path 3 study. Path 2 studies are larger than Path 1 studies and involve several hundred participants, usually 100 to 300 participants. They are usually randomized control studies, with the Path 2A studies focus on immunogenicity, dose, and scheduling finding, whereas Path 2B studies are designed as a proof of concept efficacy trials. And the next one is Path 3 trials. This aim to provide further information on safety and efficacy, which Path 3 studies are conducted on larger population and in different regions and countries, and are often the step right before new treatment is approved. Usually, it's 1,000 or more participants on the Path 2 trials. And the last one is Path 4 trials that take place after country approval, and there is a need for further testing in a wide population over a longer time frame. Where post-marketing studies are designed to provide additional assessment of rare adverse events and assess effectiveness of the new vaccine in real life and duration of vaccine-induced immunity. So the conclusion from this presentation is vaccine development is a complex multidisciplinary activity combining understanding of host pathogen interaction at the molecular level with the clinical science, population level epidemiology, and the biomechanical requirements of production. Vaccines are usually produced in small pans for Path 1 and Path 2 trials when the decision is taken to continue development of the given vaccine. A specific plan has to be built to produce enough vaccine for Path 3 trials and further. And the ultimate goal is an affordable vaccine that generates strong and lasting immunity with the fewest possible side effects, implemented without the need for expensive cold chains. And that's all for my presentation. Thank you.